Greetings everyone, and welcome to another exciting genetic engineering and biotechnology news webinar series. Our presentation today is entitled Advancing GPCR Drug Discovery Through Allosteric Modulation. I'm Jeff Bogaliskas, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. G-protein coupled receptors, or GPCRs, are a major target for drug discovery, comprising almost 30% of the current drug market, due in large part to the innumerable biological processes they mediate. However, obtaining GPCR subtype selective drugs has proven difficult, since the orthosteric site where endogenous ligands bind are highly conserved across subtypes. It has become apparent that a growing number of GPCRs possess allosteric binding sites that are topographically distinct to the location where endogenous ligands bind and are less conserved than orthosteric sites. Targeting allosteric sites with small molecule modulators has several advantages over orthosteric drug discovery, since there is a higher likelihood of achieving proper drug selectivity. Furthermore, allosteric modulators offer the potential for fine-tuning of normal physiological signaling, since they still allow for the endogenous agonist to bind to the receptor. Let's meet our panel, who will review experimental and analytical approaches for detecting and quantifying allosteric modulators, as well as how such methods can be used to reinforce efforts for understanding GPCR allostery. Our first speaker today is Nicholas Pierre, Corporate Product Manager at Cisbio Bioassays. Mr. Pierre will provide us with a brief introduction into GPCR signaling mechanisms. Mr. Pierre will be followed by Terry Kenakin, a fellow at Monash Institute of Pharmaceutical Sciences. Dr. Kenakin will discuss some of the structural properties of GPCRs as well as her data in determining allosteric sites for GPCRs. Our final speaker will be Robert Lane, Laboratory Head at the Monash Institute of Pharmaceutical Sciences. Dr. Lane will describe his functional interaction studies for GPCR allosteric modulators. Before our speakers get started, I want to encourage you to submit questions for our Q&A session at the end of the presentations. We'll try and answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. So, without any further ado, let's get our webinar underway. Nicholas, the floor is yours.
Good morning, good afternoon, good evening, everyone. Welcome to today's webinar. GPCR, they remain a major domain of pharmaceutical discovery, and they have long been an area of intense interest for myself and for all the scientists working at Cisbio. In its simplest form, GPCR signaling can be summarized as such, and it's represented on the graph here. Binding of a ligand stabilizes a conformational change, triggering the activation of one of several classes of G-protein effectors, leading to a cascade of cellular responses. Recent discoveries, however, in molecular techniques have advanced our understanding of GPCR, and it has become obvious that GPCR signaling is more complex than initially thought. Growing evidence suggests that ligand receptor binding kinetics is an overlooked factor in drug discovery, for example. Insights into the binding kinetics will advance our knowledge of the mechanisms of GPCR allosterism. Concepts of residence time, association, and dissociation rates are becoming more prevalent in the search for new drugs. To address the equilibrium and kinetic binding needs of GPCR researchers around the world, Cisbio developed Taglite. Taglite is Cisbio's non-radioactive, cell-based, no-wash technology that is quickly becoming the industry standard for studying receptor-ligand binding kinetics. The complete list of products, peer-reviewed journals describing the use of Taglite is accessible through the Cisbio website. G-proteins, they constitute one of the most important components of the cell signaling cascade. The past decade has witnessed a dramatic increase in our understanding of GPCR pharmacology, and in particular, our understanding of allosteric modulators, to which this webinar is dedicated. The discovery of functional selectivity, of bias signaling, has redefined the classical GPCR signaling paradigm. To address the growing needs for accurate G-alpha-Q pharmacology and robust screening, Cisbio developed IP1. IP1 is that assay that measures the accumulation of inositol phosphate 1 generated in response to G-alpha-Q stimulation. Through superior specificity and true assay calibration, IP1 offers tremendous advantages over the more traditional calcium flux technologies, making IP1 one of the most desirable assays for pharmacological studies. In 2015, for instance, an average of 1 peer-reviewed article was published every 2 weeks using IP1, a testimony of the research community's appreciation for the product. Cisbio cyclic AMP kits are the gold standard in cyclic AMP accumulation and can be used to study both G-alpha-S and G-alpha-I activities. The kit's high performance, their reliability, they've also been extensively reviewed in the literature and applied to all classes of GPCRs. MAP kinases are activated in response to GPCR stimulation and play essential roles in regulating receptor responses. Cisbio's ever-expanding portfolio of homogeneous assays covers all the major phosphorylation events involved in GPCR signaling. Cisbio is dedicated to the development of innovative solutions to study GPCRs. We're proud to be hosting today's webinar on allosteric modulation, and we're proud to having such talented researchers such as Mary Cannells, Rob Lane, and Terry Kenakin trust their research with our products.
Thanks, Nicholas. That was a great introduction into GPCR signaling to get our webinar started. Before we move on to Mary's presentation, I'd like to remind everyone to submit questions for our Q&A session at the end of the presentations. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All of our speakers will be available to answer your questions after the final presentation. All right, so with that, Mary, you have our attention.
So the outline of this webinar will be as follows. We will start with a brief introduction to G-protein-coupled receptors and a short discussion of the relative merits of pursuing allosteric targeting of GPCRs. We will then go through various approaches to detect, validate, and quantify allosteric drug action at GPCRs, followed by a short case study in which we have used these approaches to drive the discovery and characterization of novel allosteric modulators at the M1 acetylcholine receptor. G-protein-coupled receptors are the largest class of membrane receptors encoded by the human genome and act to transduce an array of extracellular stimuli that range from light, ions, neurotransmitters, up to peptide hormones into intracellular responses. They're involved in virtually every single physiological process, and not surprisingly, they've proved tractable drug targets. Indeed, drugs that target GPCRs account for approximately a quarter of the drugs in the clinic. GPCRs are characterized by a canonical fall of 7 transmembrane helices, connected by 3 extracellular loops, 3 intracellular loops, an extracellular N-terminus, and an intracellular C-terminus. Since the crystallization of opsin in the year 2000, the last decade has seen an explosion in GPCR biology and structural biology. And we have now crystal structures across all main GPCR families with many important drug targets represented. These data, coupled with other biophysical approaches, has given us unprecedented insights into how drugs interact with these receptors and cause the conformational changes that, for example, lead to the engagement of intracellular effector proteins, such as G proteins. As their name suggests, GPCRs couple to heterotrimeric G proteins that in turn engage a vast array of second messenger systems, such as cyclic AMP or inositol phosphates, and downstream signaling pathways, for example, the activation of intracellular kinases or gene transcription. However, GPCR signals are also tightly regulated, and the canonical view of this regulation is that following agonist activation, A GPCR is phosphorylated, for example, by G protein receptor kinases or GRKs, and then the regulatory scaffolding protein beta-arrestin binds to this phosphorylated receptor, preventing further G protein activation and recruiting the cellular machinery that is going to drive receptor endocytosis. The internalized receptor can then either be recycled back to the cell surface or degraded into lysosomes. However, more recently, it's become apparent that arrestin can act as a scaffolding protein that also engages another wave of GPCR-mediated signals. The drug discovery at GPCRs has focused on targeting the orthosteric side of these receptors, which is where the endogenous hormone or neurotransmitter might bind. And drugs can act to either mimic the action of this endogenous ligand as an agonist to activate the receptor, or block the actions of the endogenous ligand as competitive antagonists or inverse agonists. However, while such approaches have been successful, it is often a challenge to gain subtype selectivity across receptor families that bind the same agonist and whose orthosteric site is necessarily conserved. In addition, the pattern of activity of orthosteric drugs is dictated by their occupancy on the target receptor, which may be quite different from the spatiotemporal pattern of endogenous neurotransmitter. As such, there's been an increasing interest in targeting allosteric sites. These sites can be targeted by small molecules that, by stabilizing the receptor in a distinct conformation, can act to modulate the binding and action of the orthosteric ligand. This modulation can be either positive, whereby the modulator potentiates the action of the endogenous agonist, negative, or indeed neutral. And at least in theory, an allosteric approach may have a number of advantages over orthosteric targeting of GPCRs. For example, there's a chance of increased subtype selectivity because such sites may be evolutionarily less conserved across receptor subtypes. In addition, the allosteric ligand still allows the endogenous agonist to bind and act to tune up or down its action, thus maintaining the spatiotemporal pattern of the endogenous agonist signal. And finally, their effect will also be limited by the cooperativity that they exert on the orthosteric agonist, so that their effect is saturable, which might mean that they display less propensity to overdose. And driven by these potential advantages, plus the emergence of high-throughput screening approaches, examples of allosteric modulators have been discovered for all major classes of GPCRs. However, only two allosteric ligands, cinacalcet at the calcium-sensing receptor to treat hyperparathyroidism, and the antiretroviral drug maraviroc, a negative allosteric modulator of the CCR5 receptor. Now, while there is an extensive pipeline of GPCR allosteric modulators undergoing clinical evaluation, the current paucity of allosteric modulators in the clinic may result from the complexity associated with allosteric ligand pharmacology. And approaches to address this challenge will be the main focus of this seminar. And this slide really illustrates that druggable allosteric pockets have been identified across the GPCR structure, from binding within the extracellular domains, the transmembrane bundle, and the intracellular interface. So we're going to focus on the detection, the validation, and quantification of allosteric modulation at GPCR. The most direct way to validate an allosteric mode of action of a ligand at a GPCR is to determine the crystal structure of the ternary complex between the GPCR, the orthosteric ligand, and the allosteric modulator. And indeed, we have now crystal structures of all three major families of GPCRs bound to allosteric ligands. However, GPCR crystallography still remains challenging. It is low throughput and gives a static snapshot of drug action. So other approaches are needed, and at the heart of all these approaches lies analytical pharmacology. Now, we all know that GPCRs are conformationally dynamic proteins. An allosteric ligand binds to the GPCR with an affinity here denoted by Ka. Similarly, an allosteric modulator will bind to a GPCR with an affinity denoted by Kb. Both the orthosteric and the allosteric ligands can have efficacy in their own right, and this efficacy is denoted by the parameters tau A for the orthosteric ligand and tau B for the allosteric one. The allosteric modulator, indeed, can act to modulate orthosteric ligand affinity, denoted here by the cooperativity factor alpha, or the orthosteric ligand efficacy, denoted here by the cooperativity parameter beta. And we'll be referring to these terms throughout the talk, whereby values greater than 1 will indicate positive modulation, and less than 1 but greater than 0 will indicate negative allosteric modulation. Another structural approach to validate an allosteric interaction is through mutation of residues that may contribute to the binding of an allosteric ligand. However, we need to be aware that the observation of a change in the action of an allosteric modulator at a mutant receptor may be mediated both by a change in affinity, but also by a change in the cooperativity, which means that it is not necessarily a proof of a direct ligand-receptor contact, as mutations themselves can also have allosteric effects. So to distinguish these effects, careful pharmacological analysis is needed using both binding and functional assays, as well as analytical techniques. And we'll concentrate in these next. So we'll start with ligand binding experiments, which use a radio-labeled or a fluorescent-detect probe. In such experiments, the action of an allosteric modulator can be defined in terms of a ternary complex model. The orthosteric ligand A will bind to the receptor with an affinity described by Ka. And similarly, the allosteric ligand will bind to the receptor with an affinity here defined by Kb. In this case, the cooperativity between the orthosteric ligand and the modulator in terms of binding is defined by the cooperativity factor alpha. And the saturable effect of the modulator on the orthosteric ligand affinity is defined by the ratio Ka over alpha. And this effect is reciprocal. Now this model can be applied to real data. And in this light, we have the effect of a negative allosteric modulator on the binding of the orthosteric antagonist tritiated spiperon at the dopamine D2 receptor. These data can be fed to a ternary complex model to define the affinity of this allosteric modulator for the D2 receptor of about 400 nanomolar and an alpha value of 0.2. Which means that this modulator has a maximal effect of decreasing spiperon affinity by 5-fold. We can also apply this approach to competition data. And in this light here is what one would expect from a competitive interaction, which is complete displacement of the radioligand by the competing ligand. However, in the case of an allosteric modulator, the degree of potentiation or inhibition of binding of the orthosteric probe will be dependent on the cooperativity between the two. We need to bear in mind, however, that in this experiment, a ligand with high negative cooperativity will be indistinguishable from that of a competitive antagonist, as both will completely displace the binding of the orthosteric ligand. So here again is data using this approach. And we can see that the interaction between a negative allosteric modulator and tritiated spiperon at the dopamine D2 receptor. We can see incomplete displacement typical of a negative allosteric modulator, and we can pull out values of affinity and cooperativity. However, this approach requires the ligand to be labeled, and that is not always the case for the ligand that one is most interested in. For example, one might be more interested in how an allosteric modulator affects the affinity of an endogenous ligand for which no fluorescence or radiolabeled probe is available. And we can try performing a more complex experiment in which we look at the ability of our co-ligand of interest, in this case acetylcholine, to displace a tracer ligand in the absence of presence of increasing concentrations of our modulator. In both the right and in the left-hand side panels, we can see that both modulators potentiate acetylcholine's affinity. But only on the right-hand side does the modulator cause a decrease in tritiated NMS binding, consistent with a negative cooperativity with this tracer antagonist. We can apply a derivation of the ternary complex model to this data to pull out values of affinity of the allosteric modulator and its cooperativity with both the tracer, denoted as alpha prime, and also with the agonist of interest, here denoted by alpha. And finally, another approach used to detect allostery is the dissociation of a labeled allosteric ligand in the presence or absence of an allosteric modulator. And this approach assumes that the modulator, through the stabilization of a distinct conformation of the receptor, will alter the rate at which the allosteric ligand dissociates. And indeed, we can see here how two prototypical modulators of the muscarinic M2 acetylcholine receptor, alcuronium and galamine, act to slow the dissociation rate of the allosteric antagonist NMS. However, again, here it should be noted that observations of changes in dissociation kinetics are a one-way street. And if one does not observe a change in dissociation kinetics, this does not necessarily rule out an allosteric mechanisms of your compound. So, for example, if a modulator displays very high negative cooperativity with the allosteric probe, then within the range of solubility of your compound, this high negative cooperativity may prevent the formation of a ternary complex between the modulator, the tracer, and the receptor. And now Rob will move on to talk about how we can use functional assays to detect and quantify allosteric drug action behavior.
Excellent. Thanks very much, Mary. That was some really elegant work you described for determining GPCR allosteric sites. I think we're really getting some great questions coming in, so I'm encouraging the audience to keep them coming. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. And with that, let's begin the final portion of our webinar today. Rob, it's all yours.
So Mary's dealt with looking at allostery through binding experiments, but I guess what we're really interested in is how such allosteric ligands modulate receptors at the level of function, because ultimately that will dictate the physiological effect. We've already talked about the complexity of GPCR signaling, and this is now being reflected in the plethora of different approaches to measure GPCR-mediated signaling outputs. These include high-throughput methodologies such as those offered by Cisbio for the detection of a wide range of signaling events to measure, for example, second messenger generation such as an acetyl phosphate or cyclic AMP, or the activity of different kinase cascades such as the MAP kinase cascade. Selection of the appropriate assay will be somewhat dependent on your receptor of interest and perhaps your tissue or disease state of interest as well. So as we've discussed before, the identification of an ever-increasing number of GPCR modulators has been driven to a certain extent by high-throughput screening. These approaches often use the action of an EC20 concentration of orthosteric agonist, which is then potentiated by putative allosteric modulators. Such approaches, after an initial hit, are often extended in the presence of increasing concentrations of modulator to generate concentration response curves in which the relative potency and maximum effect of a modulator is derived, and that's what you can see on the slide here. While such an approach is convenient and practical to apply in a screening setting to a larger number of compounds, such an approach may miss mechanistic insights into the action of your drug of interest. For example, in this scenario, the potency of a ligand may be derived both from the affinity of a modulator for the receptor and its cooperativity with the orthosteric ligand. So you might have two different ligands with the same potency, but rather different affinities and cooperativities. So you miss that mechanistic insight. So we've taken the approach where we tend to characterize the function of our modulators in so-called interaction paradigms, in which we observe the effect of a putative modulator on a concentration response curve of our orthosteric agonist of interest. This slide illustrates some real examples and the different behaviors that were observed for positive allosteric modulators of GPCRs. So you can see in the first example, this compound 29, it both potentiates the action of acetylcholine in an IP1 assay measuring the function of a muscarinic M1 receptor, but it also acts as an allosteric agonist, and you can see that it increases the signaling in its own right. In the next example, this compound 17, what we can see, there's no allosteric agonism, but there is robust potentiation of acetylcholine. Moving on to the next example, this compound 14, what we can see is a different behavior again, where we see no potentiation really of acetylcholine, but robust allosteric agonism. And then in this last example, this is actually an example now at using a cyclic AMP assay measuring the output of a dopamine D2 receptor. What we can see is that we have both robust potentiation of agonist potency, but also agonist efficacy. So what we want to be able to do is quantify these different behaviors. And so to do this, we use an operational approach that combines the ternary complex model that we've already introduced with and that's been introduced by Mary, and we combine that with the operational model of agonism described by Black and Left back in the early 80s. And this allows descriptions of the effect of modulators on stimulus response, namely through beta, which describes modulatory action on orthosteric ligand efficacy, and tau B, which describes the efficacy of the modulator in its own right. And we still have those other parameters that Mary described, alpha, the cooperativity between the allosteric ligand and orthosteric ligand affinity, and the KB, which describes the allosteric ligand affinity. So returning then to that slide, which describes these different allosteric effects that we've observed, we can now see that we can put in actual parameters. So you can see for compound 29, it has an affinity of 13 micromole. It has this robust cooperativity with acetylcholine affinity, is denoted here by an alpha factor of 560, which means it potentiates the affinity of acetylcholine by 560-fold. And we also see robust allosteric agonism here denoted by this tau B factor of 2.5. If we then look down to the bottom right-hand side, this PAM, we can see again it has an affinity of 5 micromolar. It has moderate cooperativity with this agonist affinity of 2.5, but it has robust effects on the orthosteric agonist's efficacy, here denoted by the beta factor 12. And again, consistent with the lack of efficacy in its own right, this modulator has a tau B value of 0. Take a similar approach with negative allosteric modulators in terms of function. So on the very left-hand side, we can see behavior that we might expect from a competitive antagonist, a limitless rightward shift of a dose response curve with increasing concentrations of antagonists. In contrast, we can see that our NAM at the dopamine D2 receptor causes a limited shift of a dopamine dose response curve. We can use the same operational approach that we took on the PAMs to quantify the action of this NAM and reveal a value of affinity of 200 nanomole and a level of cooperativity, here denoted by the alpha-beta factor of 0.06, which equates to a maximal 15-fold decrease in dopamine potency. Some of you might be more familiar with quantifying antagonist action with a Schill plot. So on the right-hand side, you can see that while an antagonist, or the antagonist that we show in the example, yields a linear plot with a slope of unity, the modulator that we've used generates a curvy linear plot. However, there are examples of negative modulators of efficacy, and here is an example from our own lab that we show here. And what you can see is that this is characterized by a depression in the maximal effect of the orthosteric agonist, essentially insurmountable antagonism. We should note, though, that such effects can be observed for the action of slow dissociating orthosteric antagonists in conditions of hemiequilibrium, such as one might encounter in transient assays, such as calcium mobilization. For such observations, then, one needs further evidence to reconcile an allosteric mechanism. That might be from the binding experiments that Mary's already described, or changing the design of your functional assay. So what we tend to do is move away from transient assays such as calcium mobilization towards extended assays such as the IP1 assay that has already been described. So the last few slides have already illustrated the functional complexity that can be associated with allosteric drug action. And now we're about to make it a little bit more complicated by highlighting two interrelated phenomena, those of probe dependence and bias modulation. In simple terms, by stabilizing a distinct receptor conformation, a modulator may exert differential effects upon different orthosteric ligands. This is termed probe dependence, or it may exert differential effects upon receptor coupling to distinct intracellular pathways, effectively bias modulation. So in the top panel, we have an example of the muscarinic M2 receptor, in which the modulator is a PAM of both acetylcholine and oxytremorin M, but a NAM when the agonist probe used is anomylin. In the bottom panel, the same modulator, the muscarinic M4 receptor, displays a different level of coactivity depending on the pathway monitored. Thus, your action of your modulator can be dependent on both the orthosteric probe and the signaling pathway you observe. So to finish up, we want to show you a short example in which such an analytical approach has given us some insight into the mechanism of an allosteric modulator at the muscarinic M1 receptor, its binding site, and has also underpinned our efforts to generate novel allosteric compounds for this receptor based on this initial hit. So BQCA was first identified and characterized by, from work at Merck as a modulator of the muscarinic M1 receptor that showed absolute selectivity, subtype selectivity, over the other muscarinic receptors. We first wanted to gain some insight into its mechanism, so we looked for evidence of probe dependence. So on the left-hand side, we performed an experiment, as we sort of discussed before, where we took an EC50 concentration of various different agonists and looked at the effect of increasing concentrations of BQCA. And what you can see is that the level of potentiation depends on the efficacy of the different probe agonists. So we can see that full agonists like carbachol and acetylcholine are potentiated to a much greater extent than the partial agonists, the anomylin and pilocarpine. On the right-hand side, we took advantage of the fact that BQCA is an agopam. It also displays allosteric agonism. And we're able to completely antagonize this allosteric agonism with increasing concentrations of inverse agonists at the M1 receptor. So then this example of probe dependence seems to track exactly with or be dependent upon the efficacy of the orthosteric ligand. So it shows high coactivity, positive coactivity with full agonists, moderate positive coactivity with partial agonists, and high negative coactivity with inverse agonists. And this already gives us an inclination that this modulator is behaving to within a simple two-state model where it simply favors the active state of the receptor or stabilizes the active state of the receptor. We then extended our characterization to see if BQCA would display different effects at different signaling endpoints. We started out with measuring its effect in a GTP gamma S assay, a signaling endpoint which is very much proximal to the receptor. We observed robust potentiation of carbachol potency, but very little allosteric agonism. As we moved our characterization to progressively more amplified signaling endpoints, we could see that not only did the degree of potentiation increase, but so did the level of allosteric agonism. In fact, there was a correlation between the two, meaning that degree of potentiation tracked with the efficiency of the stimulus-response coupling. This suggests that rather than displaying bias modulation, BQCA does indeed adhere to a simple two-state model, effectively stabilizing an active conformation of the receptor. This means that not only will BQCA potentiate agonist binding, but depending on the efficiency with which the signaling endpoint is coupled to the receptor, BQCA will also display allosteric agonism in its own right. We then wanted to get some insights into where this molecule BQCA and a higher affinity derivative of this compound might be binding. So using radioligand binding experiments, along with the analytical methodologies we've described, we're able to identify residues in the extracellular domain of the muscarinic M1 receptor that contribute to both binding and transfer of cooperativity to the orthosteric site. In particular, stacking interactions with a tyrosine in extracellular loop 2 and a tryptophan at the top of TM7, so that's tyrosine 179 and tryptophan 400, were particularly important in the binding of this allosteric ligand. We're also able to apply our analytical approach to structure-activity studies. A series of BQCA derivatives were generated, and using binding and functional assays, along with these analytical methods, we're able to gain some insights into the features of BQCA that are important for the different allosteric parameters, such as cooperativity, affinity, and efficacy. We're then able to use this information, combined with some of the structural insights that we'd gained from our mutagenesis studies, to move away from the BQCA scaffold and generate a new series of muscarinic M1 receptor modulators. So interestingly, within this series, we could identify ligands with quite different properties. So on the left-hand side, we have, just like BQCA, an AGO-PAM, so it displays not only potentiation of acetylcholine, but also agonism. We have PAMs that display no agonism, and we also have allosteric agonists. This next slide illustrates how the incorporation of parameters that describe separate allosteric behaviors, such as affinity, cooperativity, and efficacy, can add texture to such structure-activity studies. And we can see that quite subtle changes in structure can have quite significant effects on a particular parameter while sparing other parameters. Such effects might be lost if one simply looks at changes in potency of a modulator to potentiate, for example, an EC20 concentration of an orthosteric agonist. So as we have seen, we can enrich structure-activity relationship studies with these different parameters that describe the action of an allosteric ligand in an in vitro system. The key next step, then, is to see how these different parameters might predict the in vivo action of such a modulator. This approach can allow us to answer some key translational questions. For example, for a particular target or a disease state, do we want a modulator of affinity, a modulator of efficacy, or indeed an allosteric agonist? Or is there a sweet spot in terms of just the amount of cooperativity between the allosteric ligand and the orthosteric ligand? The final slide was started to perhaps bridge this gap by translating our findings in model systems. So here you can see data that we've already presented in a CHO cell background at the muscarinic M1 receptor using an IP1 assay. And what we can see is that when we extend our studies into primary cortical neurons, the overall pattern of the action of these two different modulators is preserved. So BQCA, we can see in our CHO cell line. This is a low-expressing CHO cell line, so we see no allosteric agonism, but potentiation of acetylcholine, and that's reflected also in the primary cortical neurons. And then on the right-hand side, we can see that this BQZ compound not only displays modulation of orthosteric agonist potency, but also allosteric agonism in its own right, and that is also maintained in our assay of primary cortical neurons. So in summary then, I hope we've illustrated that quantitative analytical pharmacology forms an essential part of any effort to exploit allostery at GPCRs. It can give us insights into the mechanism of action of such modulators. It can enrich a structure-activity relationship with parameters that describe their action, not only in terms of affinity or potency, but also in terms of the nature and degree of their allosteric effects. Together, we can use this to bridge the gap between in vitro effects and in vivo effects, and may allow us to relate such in vitro parameters with their physiological effects. Finally, we should make two key interrelated points. At the target level, just like orthosteric ligands, allosteric ligands exert their effect by stabilizing distinct receptor conformations. Therefore, we should expect that modulators may also display intrinsic agonism, inverse agonism, or even bias modulation. Similarly, the phenomenon of probe dependence means that the nature of the allosteric effect may be dependent on the orthosteric probe used. Therefore, one must acknowledge this in your experimental design. Most likely, one would try to use the endogenous agonist, but this becomes more complicated if the GPCR is activated by multiple endogenous ligands. Finally, I just want to draw your attention to a recent review in which Terry, who will be answering the questions in the next session, is featured. And this, one of the nice things about this review is that they have a flowcharts which really go about explaining or give a pathway in how to validate allosteric mechanisms of action and how to quantify them. So we have both a flowchart for binding experiments and also a flowchart for functional experiments. So to finish, we should acknowledge the people within our own groups that have worked very hard to pull out some of the data that was shown. So for the D2 project, Chris Draper-Joyce and Herman Lim, and the M1 project, Sailesh Mistry, Alla, and Chris Choi. And we should also acknowledge the leader of the medicinal chemistry team, Pete Scammels, and our collaborator and mentor, Arthur Christopoulos. Thanks to CysBio for providing many of the assay technologies that we've used in this study, and also we should acknowledge our funding. And thank you.
That was great. Thanks, Rob. Our audience now has a better understanding of the detailed work that goes into detecting and characterizing allosteric modulators. Before we start the Q&A session, I want to remind everyone once more that this is your final chance to submit your questions for our speakers. So hurry up and send them in now. Additionally, I'd like to inform everyone that joining us for the Q&A session to help out with the questions will be a collaborator of Mary and Rob's, Dr. Terry Kenakin, who is an expert in GPCRs and professor in the Department of Pharmacology at the University of North Carolina, Chapel Hill. All right, it looks like we got a bunch of questions already, so let's begin the Q&A session. We'll try to get to as many questions as we can. So just bear with us for a few seconds while we get situated and begin the Q&A. All right, so let's get to our first question. Terry's going to handle the questions for Rob and Mary. Terry, the first question comes from one of our audience members and asks, it starts off by saying, a lot of the interaction studies are not really practical to do for an extensive drug discovery program. How would you suggest incorporating these approaches in such a program? Is there a balance between this approach and approaches that rely on looking at potency values?
Always. Good question. Of course, if one had resources, one would do the extensive studies on every compound, but you have to test every compound made by the chemist. And generally, NAMs can be treated like a standard antagonist and dealt with an IC50. First off, PAMs are a bit special. The potency of PAMs depends very much on the concentration of agonist present. For this reason, the assay that Mary and Rob described, which is the potentiation of an EC20 concentration of agonist, is probably the best first assay because it gives you an idea of the concentrations of PAM that will start to do something. and also give you some measure of what it'll do. But after that, once you have your compounds that you have an interest in, it's going to be the hard road of doing full curves in the absence and presence of a range of concentrations of the PAM or NAM, and calculating values of alpha, beta, and Kb, because those are the system-independent measures of activity.
All righty. Thank you, Terry. We have a bunch more questions for you, so get ready. The next one, our audience member would like to know, how might receptor expression levels affect the action of allosteric ligands? Should we be using low expression systems to screen for these modulators?
You could spend an hour on that question. It's quite extensive. In theory, the values of alpha and beta should not depend on expression levels. Having said that, when we start making these assays and we start to overexpress receptors, what we might do is sometimes recruit pathways as we go to higher and higher levels, and then the quality of your signal changes. Now, if your PAM or your NAM is biased, and most of them are, by that I mean they will possibly affect one signal more than another, then your quality of alpha and beta may change as well. In terms of the other part of the question, low expression's better in all cases, yes, because you're going to be able to possibly pick out alpha and beta effects. If you're at a high expression, all you'll see is a shift, and you can't figure out whether you have effects on alpha or beta, which one of those, and that can be important therapeutically.
All right, thank you, Terry. Next question is more of an application question. Audience member would like to know, what would be the application for a neutral allosteric modulators?
The honest answer is I don't know. However, there are articles coming out now that the time course of bias and various other effects can be important. In other words, if you're working out signaling, real time could matter. Having said that, I suppose a neutral allosteric compound could change the time courses where you get to these steady states. And if that has a role in therapy, then that could be important. But honestly, I really don't know of any evidence that at this point points out these are important ligands.
Alrighty, thank you, Terry. Next question for you. It starts out by saying, it seems that very subtle changes in ligand structure can have profound effects on allosteric parameters. Does this, plus all of the different effects that a modulator might have, probe dependence, bias modulation, effects on efficacy, and effects on affinity, make allosteric drug discovery much harder than orthosteric drug discovery?
This question might be from a chemist. I don't know of any theory that predicts that this should be so, but I do know that published articles and chemists and biologists I've spoken to have said this same thing. One chemist named it activity switching, where a very, very small change in structure will produce a whopping shift in what is seen. There's an article from BMS that shows subtle structures in subtle changes in structure of a single scaffold give you a NAM, neutral, and PAM just by moving a group around a ring. So this is this has been observed, I would imagine it would be an added complication in a program. The way I sum it up is allosteric research in general is a possibly more intense work. Challenges are a bit higher, but certainly much more potential reward in that you have a much wider range of therapeutic molecules that can be seen.
Alrighty. So our next question starts off by saying, Dr. Lane suggested to move away from calcium flux readout. What are the consequences of operating under hemi-equilibrium conditions?
Good question. Extremely good question. A lot of this, if you noticed from the presentation, depends upon taking data, taking curves, and fitting them to a model. Models that have equations and rules. So what's very, very important is that you get a true depiction of what's happening. Calcium transient assays do not give you that. They only give you the initial events. Much has been written about this, and so you're really not getting a correct view of what is happening often. So I can't stress enough that if you are trying to figure out how a compound works, you certainly should not use an assay that's only giving you the very earliest events of response.
All righty. Thank you, Terry. Next question. Our audience member would like to know, is it possible to create a quaternary model including beta-arrestin and different kinds of G protein?
Yes, it's always one can create a model. The problem comes in if it gets to be large or complex, then you can't verify the values of the constants that you need to separate species. Possibly the most complex I've been involved in is there's a review, and I think Rob mentioned it in his presentation that Arthur Christopoulos and I wrote, and there the complete ternary complex allosteric model is shown with the equation, and it's horrendous, and it is not useful because there's no way you could apply this and fit data. I'm thinking that if we added couplers to the existing model, we would be in danger of going there, where we have a model that would fit all kinds of curves, but we wouldn't be able to gain anything from it. Having said that, the other way to handle this question is you would have different values of alpha and beta for G protein, beta-arrestin. So if you had an assay that could just view these signals separately, then you certainly could fit different values with the existing model.
Thank you, Terry. Next question asks, what is the role of receptor oligomers on allosteric agonism, and how do you clearly dissect such an effect?
That's a loaded question. First of all, when receptors form different species like that, dimers, trimers, that's just allostery. That's just one other vector. And so the same equations apply. Having said that, I don't know of a good way that says, no, what I have is a compound is binding to a receptor to induce an allosteric event, versus I have a compound binding to a receptor that's acted upon by a different protein like RAMP or another receptor, and that's giving me an allosteric event. Again, having said that, I'm sure you could devise systems where you could only study dimers and so forth, and I've seen some of this where you create assays with Brett and Fret and so forth. But really, if you're asking the question, is there something unique in the quality of allosteric event, they really should be the same thing.
All right, thank you, Terry. Another question for you, if you're feeling up to it. Most of the basic insights into these allosteric effects come from artificial cell systems with overexpression of the target of interest. How do these findings translate into an in vivo situation, and how can one measure these?
Yes, very good. Well, I'm going to take your question and drill down into it a bit. Recombinant systems can be extremely useful. Why do I say that? Because we all know that you could be critical of that. They may give you events that aren't going to be seen in a natural system. When I worked in industry, we felt that our job as biologists was to characterize the compounds that our chemists made. And so your job is to reveal what your molecule can do. You're not really sure that it's what it will do, because that'll depend on the system and cell type and so forth, or it can. But at least you can use a system that you engineer and say this molecule is going to promote interaction of the receptor and GI protein. So you can define that. The other part of your question, however, said overexpression, and if you mean receptor, you're asking for trouble, because you're going to possibly get a complex signal that will be hard to work with. So recombinant systems can reveal what your scaffold can do, but I wouldn't necessarily overexpress things.
All right, Terry. Looks like we have a technical question. One of our audience members would like to know that they say that they're encountering difficulties in building in GraphPad Prism 5.0 the equation of the operational model of allosterism. They would like to know if there's any template or guide available for assisting in building this equation.
This question's easy to answer. Call Arthur, Rob, and Mary. No, it's pretty easy to program equations in Prism, and if you're seeing any changes in maximal response, the equation in there doesn't handle that. There are good reasons for that, but you would have to program the equation in, and my suggestion would be to contact someone who might be able to help you if you're not into doing that. But it's fairly easy. If you can program Excel, then you can program Prism.
All right, thanks, Terry. Next question for you. It says, in order to use the operational model of allosterism equation, must the modulator KB value be introduced and previously attained from radioligand binding assays? And in case that is yes, can radioligand from different orthosteric agonists be used?
Yeah, okay. That's a great question, and it opens up a important topic. I'm generally not in favor of taking numbers from a binding assay and applying them to an allosteric system, simply because allosteric systems are cooperative, and so all your real numbers that matter often depend on what else binds to the receptor. Having said that, NAMs, the effect is minimal. So I'm going to say, yeah, with a NAM, you get a fairly useful answer that you could apply, because alphas are generally small, betas are small, and the observed Kb is the true Kb divided by 1 plus concentration of ligand divided by that times alpha and beta. So PAMs, however, don't do it. PAMs really, really depend on what else is binding. So the answer you get, what happens if you read articles on PAMs and they use the equations, you will see Kb values that are way lower than the concentrations that start to do anything. And you say, well, how can that be? The compound isn't even binding. But it is, because that Kb is just the raw Kb of the compound binding to the protein. As soon as you bind something else, that Kb goes sky high in terms of times alpha and beta. So binding, not useful for PAMs, marginally useful if you must with NAMs.
Alrighty, thank you, Terry. It looks like we have maybe time for one more question here. Staying on the same topic, our audience member would like to know, what is better to use to measure PAMs and NAMs? Stable line made with your detection or a transient? Can we use heterologous cells?
Well, that's as I pointed out earlier, as long as you're viewing a single signal, say you've made a cell line and you've engineered it such that you can see beta-arrestin, then you should be getting answers that apply only to that signal. The problem comes when you're trying to think about whole cell response, because we now know, because we can view signaling with different assays, that efficacy, which used to be considered a cell signal that only had differentiation in terms of strength, quantity, now it's got quality. You have biased ligands that produce different qualities of efficacy due to different mixes of signaling to the cell. Well, allosteric molecules will change that. So you can use your natural hormone or transmitter, potentiate it, and induce bias. This is well known. And if you're using heterologous cells or different cell types, you could conceive of where the answers you get in terms of different pathways and bias will change. But if you can get a simple readout of a single signal, you should be okay with any kind of assay.
All right, thank you very much, Terry. That was excellent. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on our website at www.genengnews.com for 1 year. So if you missed parts of it, you can watch it again, or feel free to forward the link to your friends and colleagues, which we always recommend. I'd like to thank Nicholas, Mary, Rob, and Terry again for their informative presentations and answers. And I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to SysBio for sponsoring this webinar. So hopefully we'll see you again in another Gen webinar in the near future. Goodbye for now.